Good morning, everybody. I'm Bernard Saggaert. I'm the COO and VP Manufacturing of the company eTheRNA, located in Belgium. And I'm going to talk to you about the production of RNA, the winding road to GMP, the things that you need to decide on if you want to go that pathway, and also give you our experience to do this during a COVID-19 pandemic, and which impacts that this was associated with for us. Maybe just first some words around eTheRNA, some highlights. We are one of the big founders of the RNA technology, next to the big ones that you all know. We have been active since 2013 as a company in the RNA field, but actually I have people in eTheRNA that have more than 30 years of experience in RNA production and processes. We, as a company, we focus ourselves to do the development of products. We focus ourselves on 4 technology pillars, being RNA chemistry, antigen identification, novel lipids, and process engineering, but we'll come to that later. What makes us a bit specific as a company is that we have an integrated GMP manufacturing suite, and that we therefore can design our processes ourselves, which we also do. And that based on the 4 technology pillars, we actually make products in target areas as oncology and infectious diseases, but also thinking about autoimmune, CNS, and rare diseases. As I indicated, I have persons in employment that have 30 years of experience in RNA. And since we are a spin out of the VUB, we have a solid academic foundations. And a very experienced management team with lots of scientific expertise, clinical development expertise, and also manufacturing know-how. Looking at the history of the company, back in the 2012, before actually the company was really founded, all of the work was done at the VUB in Brussels, Belgium, and there we did the first preclinical and also clinical trials to substantiate what we are doing now. In 2013, we spun out of the VUB as a company and were founded in a Series A funding event. And at that time, we had around 35 FTEs and 2 patent families. And if you look at where we are now, and we are at the end of that period, because the period that we look at was 17 to 21, where we were funded in a Series B funding of 39 million. In that time, we grow to 75 FTEs. We have multiple patent families. So there's really big steps forward that we did during that period. We also established a GMP facility, and we also made new processes available for our products. Towards the future, we will expand further in other areas than the ones that we are currently present. We will also expand into other RNA modalities like self-amplifying RNA or circular RNA, and continue to work on formulations. And the intention is there to continue growing and do 2 new INDs per year and continue to file IP. This has led to this therapeutic areas where we already see that we have different products and different products in the pipeline. The one that we brought to GMP and also the first in human is the A002. It's actually a head and neck cancer product, which is based on multiple RNAs compiled in one LNP. And that's the one that we brought together with partners to the clinic, and the first in human will happen in the beginning of 22. You can see that we have multiple other projects running, both in oncology and infectious disease, and what we want to do towards the future. Focusing more on the things that are important for manufacturing, because this is still the topic of today. The 4 pillars that we're working on is actually the 1, 3, and 4 are the ones that are important for manufacturing, since the 3 is more linked to a disease area and not to production. First, in the field of RNA chemistry, one of the things that is important there is optimize your constructs for manufacturing, but also for expression. We'll go a little bit further into that. Then lipid chemistry is anything around LNPs and the formulations that are used to administer RNA. As you potentially know, RNA needs to be protected from ourselves, humans, because we have RNases in our body and on our skin, which can destroy completely RNA. So you cannot inject or use RNA as a non-protected product. And then in the area of process engineering, what is typically important there is the facilities that you use, the processes that you use for drug substances, the processes that you use for drug products, but also things that we are working on towards the future. Like, for instance, we have an engineering program running with a partner where we want to design the RNA processing equipment for the future. Another thing that is in that area of process and engineering is anything around thermostability, where we look at, for instance, lyophilization to improve thermostability of products. A first point to touch upon is facilities. What do we have in eTheRNA as facilities? We of course have development labs, where we do plasmid development, small-scale plasmid production, small-scale research-grade production. And also lots of work is being done on the development of the processes, where a large focus is given on the upstream part, because it's my idea that anything that you do not produce as contaminant or side product, you don't need to remove in your downstream process. And that's why we focus on upstream, but in the end, you need to remove anyhow proteins from your enzymes. So you need to focus on your downstream as well. And a large field that still has to be explored, and then we're putting lots of effort in there, is the QC development of methods that are needed to support whether product release, but also the development of the processes. Next to that, we have in that NEAL 1 facility, the upstream processing rooms, which are currently still aseptic, but we also have the bioburden reduction included in the process, and we also can do sterile filtration. A focus needs to be on RNases and DNase-free processing, not to destroy your products. We do in that NEAL 1 facility mainly the upstream part, but also a little bit fill finish and sterile filtration. But products after the IVT move to the NEAL 2 facility, where we do the our semi-automatic downstream purification and bioburden reduction filtration. I also have a team in Ghent that is sitting next to the discovery team, and that is working on formulation together with the discovery team. As starting points for RNA manufacturing and also bringing RNA manufacturing to GMP, there are some things that you think of, and the first thing in there is what we call RNA chemistry, and that is construct optimization. Constructs you need to optimize them for two reasons. One is you want to see what to increase as much as possible the expression of your RNA in the cells, and you can do that in different ways. One of the things that is at your availability is optimizing the codons in the open reading frame, but you can also optimize your UTRs, your untranslated regions, the 5' and the 3', to express, to have a better expression in cells and also as a more stable RNA. Something you need to think about from a regulatory point of view is which antibiotic resistance you're using. Because in some countries, some antibiotic resistance genes are not allowed or not preferred anymore. One of the other things that you need to think of is which restriction sites you use, because your restriction sites can also be present by accident or if not chosen correctly in your open reading frame, which would mean that you're during the linearization, where the restriction is applied, actually your construct will break into pieces. The next step is within the manufacturing method of your plasmid. How will you have it manufactured? Will you use PCR techniques that bring you to linearized DNA template, or will you go over the pathway of making first the circular plasmids in E. coli and work from there? That's a decision that you need to make. And then also you need to take decisions on the different grades that are available of plasmids. You can go early on with research grade material, but at a certain point, you will at least have to go to high quality, and for certain countries to GMP quality of plasmid to go into the clinic. If you go to GMP or high quality, you also need to decide what your requirements are for master cell banking and working cell banking. On this slide, you have a typology of how a typical RNA strain looks like, with that first A cap, then this 5' UTR region, then your open reading frame with different components that can be included there. We include, for instance, a DC lamp, because we target mainly DC dendritic cells. After that, you have your 3' UTR, and then your poly-A tail. That's a typical construct, RNA construct. That is how it looks like. If you look at the other starting materials, similar things need to be decided upon. From a production point of view, you need to see which materials are available and which grade is available. Lots of the material that is currently used in the RNA manufacture does not have a supplier that can supply GMP grade material or fully characterized materials. So you need to take decisions there and do risk assessments on what type of material you can use. You also need to take a decision on which process and which materials you will use, like for instance, which capping reagents are you using? Are you going for enzymatic capping, or are you going for co-transcriptional capping? Which NTPs do you use? Do you use modified NTPs, regular wild type NTPs? The choice of enzymes for the different steps in your reaction need to be decided upon. And also the quality level that is available and that you can use. Next to that, you also need to take decisions on the quality control of those materials. Do you work with initially with suppliers, CFA, and is that sufficient, or do you do yourself some release test or some characterization test to make a selection of materials? Moving from the starting materials then to the drug substance. a typical process for a drug substance production, and then mainly if it's plasmid based, because that's the one that we use, the steps that you execute are typically a linearization of your plasmid DNA. If you start with circular DNA, you linearize it, you purify your linearized plasmid, and you start your IVT reaction. Then depending if your capping is done co-transcriptionally or post-transcriptionally, you need to do capping. The same for polyadenylation. If your construct did not contain any poly-A tail yet, you can do the polyadenylation afterwards. After which you need to purify and do a bioburden reduction filtration. Depending on use, you need to also do a sterile filtration, and then after that, you need to go to your final concentration that you need in your drug product production process, do a concentration adjustment, and do a filling operation depending on its use as well, bulk or individualized. That's based on your uses. The same as for raw materials here, you need to decide which release test you will apply to, which characterization test you need, that need to evolve further into release tests, and also by when you start to do stability studies. These QCs can be largely stage appropriate or stage depending the further you go into your process, and that's not different than with other biologics that you develop. You need to have a plan, a development plan for your QC tests to indicate where what is needed at what stage and what you need early on to support your process development as well. So if we now see how we can bring a drug substance to GMP in such a process, this slide gives you, I think, a good overview on how you need to approach all of this. And also, it's not only bringing the production to GMP, it's also bringing your product to GMP. And therefore, you have different phases in your product development. And for us, the roadway to GMP does not start at the moment that you say we need to bring this product to GMP. It actually starts a lot earlier, because it starts in R&D and discovery. That's there that you first have your products made, research grade products made. That's there that you do your UTR optimization, your codon optimizations. You start with small scale proof of concept productions, where if you make multiple constructs, you also can do in vitro construct screening to get to your best drug, your best construct to bring to GMP. And it's only then that you start to make some small scale still, but it's a bit larger scale batches that are already based on a process that can be brought to GMP, because the early stage milligram scale proof of concept productions are based on a downstream process that is totally not optimized to bring to GMP. And once you have those smaller scales done and you already started your QC testing and maybe already QC development, you can start thinking about non-GLP batches, GLP batches that can be made. Whether or not they are combined with engineering batches or not, that's debatable, but sometimes it can be done if timing is right. And then you move to verification batches. As I already indicated, construct-dependent QC method development needs to start as soon as possible, because they are decisive on process decisions that need to be taken. At a certain point, your QC methods will have to be validated. Your processes will have to be validated if you go more into clinical phase 2. And well, our advice is definitely to start stability studies as early as possible. At a certain moment, you might also consider to start working on the requirements for a tech transfer if really large quantities would be needed, and you need to have, like it is for COVID vaccines, you need to start producing for a global pandemic, for instance. Okay, that was a little technical error. If you look at the drug product requirements, then this is typical now for lipid nanoparticle products like the pandemic, the vaccines for the pandemic from Moderna and BioNTech. You need to decide here production-wise which LNP production method you will use. There are some products or methods available on the market for LNP production. You need to decide on a storage buffer where you put your LNP in, and that's really an important one because it always also will determine partly your thermostability. You need to do your sterile filtration, you need to do your fill finish, and also decide on your fill finish volume, multi-dose, single dose, these type of things need to be decided upon. The same thing as for drug substance counts also for drug product. You need to determine your appropriate QCs for the stage you're in, and that both release, characterization, and for stability studies. A similar process is actually true time-wise for drug product as for drug substance, where you early on need to decide on your lipids, which lipids you will use, which ratios you will use for the lipids. That's really an important decision because it really determines all the characteristics of your LNP. But you also have to think about a method to do the manufacturability assessment and early on look at stability and QC development. And then the pathway is actually completely aligned with what you do with drug substance, which is not surprising since both need to be incorporated in the same product. So the flow there is really identical. With regards of which regulatory is applicable, there are some differences between the EU and the US, but it's mainly in the details of the guidance, since both sides of the ocean have determinations like vaccines, but also advanced technological medicinal products on EU side, and then cellular and gene therapy, it's called on US side. The legislation is different, but on the other hand, the content is relatively similar. For phase 1, you have some differences in what can be, what is needed for GMP manufacturing, where in Europe, your manufacturer does need to have a license and does need to be inspected, specifically your drug product manufacturer, and that in the US is not really the case. In this route to GMP during the pandemic, if we look back and we assess what the pandemic has brought us compared to what would have been there in normal times, there are some things that were really impacted. And definitely one of the things that is still impacted, and hopefully it will be resolved one day, is the supply of single-use materials. Our complete manufacturing method and process is based on single-use materials. So the impact of having long lead times makes us a lot less flexible than we were before. Lead times have extended dramatically and more than doubled, which means that we have to already get a better planning from discovery and the other departments of the company, and looking forward what needs we would have, and really plan everything out. Some of the raw materials have also prolonged lead times. QA has been impacted because audits needed to happen remotely due to the impossibility of travel, which is something that we had to learn, and also companies had to learn, because doing an audit without seeing a process, without seeing a facility, is really difficult. We solved it, and suppliers have solved it by showing off movies, but like anybody in a movie, you like to look at your prettiest. So it's more difficult to see through things than before. There was, for us specifically, a technology transfer that needed to be done from one supplier to another. And all of that has been done site to site in a time zone that was definitely not our time zone. So that made it really difficult to do technology transfers in such a way. And a bit the same happened with QC development. Everything needs to happen remotely, which doesn't make it really easy to do. But in the end, we've coped with it, and that's also why our product is the development of the processes is finalized, and we just need to make 2 batches, and that will be in that way we will be fully clinically ready. Things that specifically impacted the working during the pandemic is certain things that happen beyond your control, and definitely the US Defense Act was one that privileged certain US customers from US suppliers, and that made it really difficult for us to get access to certain materials. But that also meant that from the moment that we identified that we started to look for alternatives, and that we are now, because of those alternatives, we are better prepared towards the future. Another one like that is transportation. If there's less airplanes in the sky flying people all over the world, there's less room for materials in airplanes. So this means that it made it more difficult to transport materials. There are some materials that had a high demand specifically for the COVID vaccine productions. And I think also there the US Defense Act didn't privilege European customers. And then local travel regulations impacted us as well. One of the things that was less impacting than initially thought was a reduced presence on site in the production site. Having less people on site did not really impact us in a way, because well, the production could happen in the clean rooms. People are sufficiently gowned to be protected from each other. There's also very high ventilation in those rooms, so the chance of being of contaminating somebody in such a room is really minor. The only measures that we had on site was that management was less on site, and also if work could be done remotely, people were instructed to work remotely, so that the people that were on site were as much as possible protected. Another impact was also contractors that need to come on site, that at first they were not allowed by their management to come on site, which meant that some technical or maintenance work was delayed. But in the end, that did not affect us too much. So all by all, I think we have been impacted by COVID, but the impact was minor. And the main impact was specifically around one of the materials that was really delayed in supply, and that had to come from the US. That was the major impact that we had during this period. This is at the end of my presentation. I hope it was interesting, and I'm open for some Q&As afterwards. Thank you.